Hello, everybody. Welcome to today's webinar, which is entitled The Last Exit Before NGT and GMM Are Getting Out of Control. Thank you all for being here. This webinar is going to be recorded, the first part of it, which is the different speeches. But then the part where we're going to discuss with you is not going to be recorded, so everybody can freely express themselves. And you will be able to hand in questions using the question and answer tool, which you find at the bottom of the screen. And there will be time for discussion after each speech, but also at the end of the first part and at the end of the second part. So the first part is going to deal with plants derived from new genomic techniques. And the second part will deal with microorganisms derived from new genomic techniques. I'm just making sure that everybody has received the updated program because there were updates at one moment. So I'm pasting it for you in the chat. And this webinar, of course, comes at a very crucial moment in time because, as you know, beginning of next week, we have votes foreseen on both NGTs and GMM. So there's a vote in the ENVI Committee of the European Parliament on NGTs on Monday and a plenary vote on Wednesday. And then on Tuesday, we have a vote at council level on the microorganisms. So it's really quite a full week, and we are wanting to make sure that everybody has received the information of what would be the consequences, especially for environment and nature protection, if these proposals go through as they now stand. As you may be aware, if you have followed our work, we have been criticizing the proposal on NGT plants for being based on unscientific criteria, or to put it more blatantly, the Commission has tried to give itself a pseudo-scientific facade by basing itself on the opinions of EFSA, but there are many criticisms towards these opinions, and in our opinion, they are not sufficiently based on science. But I will let my colleague Andreas Bauer-Panskus tell you more about that. Andreas, we've had a recent letter exchange with EFSA on precisely this subject. Could you please fill us in?
Hello, everybody, and welcome. So I will give you a short introduction on today's subject in overall with EFSA specifics and try to set the stage for my colleagues, which will elaborate more on all these things in detail. So thank you, Mario, for sharing the slides. So the proposed regulation on NGT plants, how legal and scientific arguments got screwed up. Next slide. The starting point is the decision of the European Court of Justice in its 2018 decision, which you all know, of course. The court clarified that plants obtained from new genetic engineering or new genomic techniques, or NGTs, however you will name them, have to be regulated, as the risks can be similar to those of transgenic plants, and they have no history of safe use. Next slide, please. In the course of the political and regulatory process, EFSA was commissioned to produce several scientific opinions on this subject. The outcome are several reports about the risks of NGT plants. However, unlike several national authorities, like from France or Germany and other institutions, EFSA has never presented a systematic analysis of the differences between NGTs and conventional breeding. Next slide, please. Instead, EFSA is searching for so-called new risks. According to their publications, they could not find such risks. However, there is no clear definition of what these new risks are meant to be. They may concern risks in comparison to what is known from conventional breeding or in comparison to what is known from transgenic plants. These differences matter. Next slide. So, concerning the difference to conventional breeding, if EFSA would seek for examples of NGT plants that are new to the environment, there is a large number of publications showing that risks of many NGT1 plants go far beyond what is known from conventional breeding and what is known as safe use. EFSA failed to address these findings. Next slide. Concerning the similarities to transgenic plants, if EFSA were to look for risks in comparison to those already known from transgenic plants, there are several examples for NGT1 plants. It is not relevant if these risks are new. So, should be a no-brainer if NGT plants have similar risks as transgenic plants, they have to be regulated. Next slide. So to clarify these things, we wrote a couple of letters to EFSA. There's a correspondence starting in March this year to May of this year. You can find all the letters if you follow the link below. And in all these letters, the authority did not bring any arguments to contradict the findings of Testbiotech. So from a scientific point of view, the whole approach of EFSA is quite shaky and gives the impression that EFSA tries to avoid a meaningful discussion about environmental risks of NGT plants, thereby undermining the European Court of Justice ruling. Next slide, please. Overall, the findings of EFSA are in contradiction to many scientific studies regarding risk potentials of NGT plants. These include negative effects on pollinators and food webs, invasiveness, weakening of natural plant populations, yield depression, insect toxicity, and effects on soil organisms. You can find references for all these effects in our short publication. You find the link below. Next slide, please. In a very recently published paper, also from 2026, co-authored by a former EFSA GMO panel member, Niels Rostocks, on environmental risks of NGT plants like oilseed rape and potato. Next slide, please. The authors come to the following conclusion. Given the unpredictability associated with altered phenotypes or enhanced fitness in oilseed rape and other brassica species, such varieties should be subject to rigorous case-by-case biosafety assessments. Overall, sustained research on the interactions between GM plants and NGT plants and their wild relatives is crucial to ensure informed, scientifically sound evaluations of environmental risk and biosafety. Next slide, please. So the conclusion of my short talk, the EFSA approach leads to a systematic underestimation of the risks of NGT plants. Most crucial elements, such as the equation of conventional breeding with NGT1 plants, are not backed by any scientific justification. Also, the differences between the risks of NGT1 and NGT2 plants or transgenic plants remain largely undefined. Next slide, please. Therefore, we recommend to reject the current proposal. At the very, very least, we demand the adoption of the following amendments. Next slide, please. Apart from the rejection of the Council position, specific safety criteria should be imposed on NGT plants which can spread in the environment. Detection methods are essential for traceability, monitoring, and labeling. Also, there have to be criteria for a withdrawal of NGT plants from the market in the case of health and environmental problems. Next slide, please. And next slide. Next slide. Last but not least, the traits that are excluded from NGT1 status, like tolerance to herbicides and the production of a known insecticidal substance, have to be amended with plants that are able to spread in the environment. So that should be all for the moment. Thank you very much for your attention.
Maybe Andreas, could you show the last slide again, because I think it's so everybody can see the numbers of the amendments which you want to introduce. So the slide before was just shown very slide. So these are the other amendments which were explained but not shown. Thank you very much.
Yes, thank you. Thanks, Andreas. And for the numbers of the amendments, we can, of course, communicate our recommendations, and we will also communicate our recommendations to everybody concerned. And then you have it also in writing. I think it was made very clear from your side what is the problem with the scientific base. Let me see if there are questions. All right. There are no questions for the moment. Just people cannot access the program for the moment, it seems. Let me try to post it for everybody again. Okay. Anyways, if there are no questions for the moment, then I think...
It may be that the chat is disabled for the public. Maybe you should post it in...
I tried it in a different way now. I tried to send it in a different way now. So let me know, please, if it works. If there are no questions for the moment, you can still ask questions to Andreas at the end of the first session. And then I would propose that we go... Oh, now I see that the questions are coming in. So there's one question. Can I ask if your last proposal on plants that can reproduce, is this to capture all crops as well? Just trying to understand the scope. I think the answer is yes, right, Andreas?
Yes.
Yeah.
I have to read it again. The last proposal on plants that can reproduce, is it to capture? Yes, of course.
Yes. There could be crops that have wild relatives, for example, and so on. All right. Well, I propose we continue with Christoph. As I said, I think, Andreas, you have made it very clear what are the problems with the scientific base. But now the question is, of course, what does this mean in practice? If we base our legislation on such a questionable scientific basis, what does it mean in terms of environmental risks? And there I think Christoph will give us an update on the latest developments in research and show us some examples of plants that could pose an environmental risk if the legislation as it is now goes through. Please, Christoph, you have the floor.
Yeah, I tried to give one example for recent research which shows that the current proposal is not fit for purpose. And after that, the risks will be presented in more detail by colleague Franziska Koller. So this proposal deals with NGT plants that show stronger effects as caused by transgenes, but no mandatory risk assessment would be necessary. Next slide. So the general issue behind is changing the gene expression of the plant-owned genes. So no additional genes are inserted, but the activation of the genes is enhanced. The gene activity is enhanced by NGT methods, and so there are especially new CRISPR gene scissors that allow the introduction of small but complex changes within the regulatory elements of the genome, such as promoters, for example. Typically, these changes cannot be achieved by previous methods. And these changes then can cause effects that go beyond the known characteristics of the species just by changing the gene activity of the plant's own genes. And of course, the risk then will be dependent on the functions that are changed. However, such plants typically would be in the category of NGT1 plants with no mandatory environmental risk assessment necessary. And next slide. There is a recent publication from 2026 which deals with so-called hypermorphing. And hypermorphing is an expression that is chosen by the authors themselves. It comes from Greek language. It means a high level of potential of altering organisms. And the authors belong to the team of Jennifer Doudna, which is one of the inventors of the gene scissors. The institute is called the Innovative Genomics Institute, and most of the authors belong to this institute of Jennifer Doudna that was started up at the University of California. So what is this about now? Next slide. This is according to the outcome of this publication. It's a new dimension for NGT1 categories, which is based on smallest changes but biggest impact. So the general strategy behind hypermorphing is to introduce really very small changes, the minimum of changes in regulatory elements of the plant genome, but changes which then result in maximal hypermorphic effects. And the scientists note very well that these genetic changes could not be achieved by previous methods. And when you go to the next slide, the general strategy is shown in this figure, which is adapted from the publication. So it's a combination of these small changes in the regulatory elements, such as promoters, and to maximize then gene activity, and the resulting genetic changes are then called hypermorphic. And this is exactly what the authors were seeking for. Next slide. So this shows the method again. It's not only based on NGT technology as such, introducing the gene scissors into the plant material, but it's based also on an automated process for the assessment of the resulting effects. So it's a three-step process, one could say, which includes the genetic engineering process, then the automated process for effect quantification, and then this is repeated in circles to improve the outcome. And the outcome is shown in the next slide. It's symbolized by this plant. So what was really achieved in these examples, there were millions of biological samples being explored, tested with a wide range of CRISPR-made changes, and then the effects were assessed by automated processes, and the outcomes led to a 30-fold increase in gene activation. This is really impressive, a powerful technology. In this case, it was targeting a gene that are related to photosynthesis and protoplast of plant species of sorghum, but the method is in no way restricted to any species or target genes. So this is really something which never would be achievable by conventional breeding, and this goes beyond. So now, next slide, we see a quote from the publication from the authors themselves, which say, within these regions, we identify compact deletions and motif insertions that strongly increase protein production, 30-fold relative to the wild type increase, outperforming transgenic enhancer elements. So very small changes, but somehow complex also, nevertheless. Several low number of changes was introduced, but in a specific combination, these changes then led to this high effect on the gene activity in these plants, and this goes even beyond what we see from transgenic elements. So this is the recapture of this publication, and now next slide. Of course, we could discuss the risks, but of course, as said before, these will always depend on the target genes and the plant species, like impact on pollinators, protected species, or food webs, or the plant-soil interactions, plant health, or increased invasive potential. All these issues then would be an issue for in-depth risk assessment, and in the following presentations, you will get an idea about the potential impact on pollinators of other plants. But indeed, there is something which has to be risk assessed before these plants could go into the environment. But now when it comes to the situation in the European Union, there is on the one hand something what one could call the challenge posed by the European Court of Justice, which says at least those plants with risks that are similar to those as transgenic plants have to undergo mandatory environmental risk assessment. And in short, you could explain it in much more words and much more details. Next slide. But when it comes to now to the specific example, this NGT hypermorphing has no history of safe use. The effects achieved go beyond transgenesis even. However, according to this NGT proposal is now on the table, such plants would categorize as NGT1 plants because the number and type of genetic changes to cause these effects are below the threshold proposed by the new legislation. Thus, the legal challenges posed by the court are not fulfilled by this proposed legislation. Simply spoken, you cannot solve the challenge as posed by the European Court of Justice by just introducing a magic threshold of 20 mutations. This concept is not based on science. It's largely irrational. It's born by bureaucrats that try maybe to circumvent legal requirements by disregarding science, but it's not in line with many publications and the opinions of many experts. Next slide. So maybe one could say there is even a very basic question behind this discussion. The EU has to take a fundamental decision whether future NGT regulation will be based on science or on post-factual ideologies. But in any way, next slide, if the Parliament does not want plants obtained from NGT hypermorphing to be released without environmental risk assessment, then it has to say simply no to the proposed regulation. Thank you very much. Further informations you can pick up in other publications. And also we have a news item on our website on this hypermorphing publication. Thank you very much.
Yes, thank you, Christoph, for this overview of the latest scientific developments. For the moment, I don't see questions, but we can do the discussion at the end of this first session. I think that will be very good as well. And in that case, we can continue with Dr. Franziska Koller, who will then fill us in on examples of NGT plants that can have impacts on pollinators. Please, you go ahead.
Thank you, Astrid. I hope you see the slides now.
Yes, thank you.
Yes, I will give a first outlook on potential impacts of NGT crops on pollinator in case of long-term and large-scale cultivation. First, about the present situation of crops and pollinators. Population of insects, including pollinating insects, have been declining for decades. And this is due to a combination of factors, including poor nutrition, agrochemicals, and diseases. And the poor nutrition of pollinators is caused by limited nutrients, for example, in agroecosystems with monocultures and reduced diversity of flowering plants. However, flowering crops can support pollinators by providing pollen and nectar, and especially mass flowering crops can have strong positive effects on pollinators, particularly during periods of when only few flowering plants are available. Many crop species are pollinated by insects, and their harvests also depend on pollination. Examples of pollinating insects are, of course, the domesticated honeybee, as well as wild bee species and other non-bee taxa like beetles. We were now interested if currently developed NGT crops may alter the relationship between insects and crops, and whether they could further worsen the situation for pollinators. Therefore, we did an horizon scan to identify relevant NGT crops. We concentrated on insect-pollinated crops, which are cultivated in large scale in Europe. This is, for example, oilseed rape, maize, grapevine, apple, and so on. And we then scanned NGT applications in these crops and focused on market-relevant examples in current scientific literature. We identified in the last steps NGT alterations in pollinator-related traits. So crops that exhibit modifications in pollinator-related traits. These could be alterations in trait with impact on plant-pollinator communication. For example, the scent that a flower emits to attract a pollinator, or alterations in traits with impact on pollinator nutrition and health. For example, beneficial compounds for pollinators in the pollen, or alterations in traits with impact on plant pollinator temporal match, which means that a flower of a plant and the respective pollinator occur at the same time so that an interaction can occur. We scanned for direct effects of the NGT application and also indirect and unintended effects. I already mentioned some of the interaction between plants and pollinators. And here we can see now a flowering plant which emits various signals such as color and scents. And these signals can, on the one hand, help in the plant defense against pathogens and predators. and on the other hand, they can attract pollinating insects. Hormones that are active in the plant regulate various processes in the plant and also some of the key traits in pollinator interaction and attraction. For example, the color of a flower is regulated by plant hormones as well as the nectar composition. And further, the plant hormones have an important role in the timing of the emission of scents, the nectar secretion, and also the initiation of flowering. We have found now diverse NGT applications altering the plant hormones. For example, the synthesis of a specific hormone is reduced. These applications often have the aim to increase plant resistance against, for example, stress like drought. But given the role of hormones in the interaction between plant and pollinators, it can be assumed that the manipulation in the plant hormones may, beside the intended effect, also affect established plant-pollinator interactions. It was shown in the past, for example, in transgenic plants, that manipulated jasmonic acid or auxin to plant hormones can, for example, change the scent of a flower or the nectar quality. This can in turn lower the attractiveness of a flower, and an insect is not able anymore or interested to find and pollinate the crop. There could be also effects on the nutrition of the insects if the nectar composition has changed, for example. And further, the timing of scent emission and flowering can alter, and this can lead to temporal mismatches. It is very complex to predict what effects altered hormone signaling will have on plant-pollinator interaction in each case. But however, it seems likely that the effects caused by NGTs can have impact on established plant-pollinator interactions. We next looked at secondary metabolites. These are plant compounds that shape the scent and the color, and they are therefore highly important for plant-pollinator interaction. In addition, secondary metabolites are also part of the diet of pollinator, which consume these secondary metabolites when they eat the pollen of the plant. And for various secondary metabolites, there were shown positive effects on the pollinator health. We also found here diverse NGT plants where the content of secondary metabolites is altered. And similar to the case of altered hormones, altered content of secondary metabolites can alter the scent of a flower, and therefore also the attractiveness of a plant can be altered. In the past, it was, for example, shown that even a minor change on DNA level can result in altered color, and further, they showed that this lowered the attraction for established pollinator and lead to a pollinator switch. In addition, we assume that a reduced content of specific secondary metabolites will have an effect on pollinator health. This is due to the positive effects I already mentioned of these compounds for pollinator. For example, improved disease resistance and increased pesticide tolerance in the pollinator. I will not go into detail, but similar effects on bee health are to be expected in cases where the oil composition or fatty acid composition is changed, especially in plants used for vegetable oil and fuel, like oilseed rape developed with altered oil composition. And this will probably also influence the oil content or fatty acid content in the pollen. But these dietary fatty acids have various roles, for example, in brain function and reproduction of pollinators. We identified another field with increasing NGT approaches, which may have an effect on pollinators, the alteration of flowering time. For example, a longer growth phase and later flowering are expected to result in more robust plants with higher yields for some crops. But there are also various approaches aiming earlier flowering. In a worst case, a shift in the timing of flowering leads to a temporal mismatch, so that a pollinator and open flower do not occur at the same time point. Effects like these are also described in the context of climate change, and a shift in the timing of flowering could extend periods of low flower availability. And this can then have consequences for the pollinator, which may rely on the crop itself for pollen and nectar, for example, in semi-natural landscapes. To sum this up so far, the alteration of primary and secondary compounds, as well as plant hormones using NGT, may have consequences for the pollinator when the respective change is also apparent in the floral organs. In addition, the altered flowering time can lead to temporal mismatches. We identified furthermore indirect consequences that I will just shortly mention. First, we often see NGT modifications that decrease plant defense compounds. These repel pest insects or attract natural enemies of these pest insects, and therefore reduced plant defense compounds increase problems with these insect pests, and in result, this could lead to increased pesticide use, which is a major stressor for pollinators. Further, NGT crops with increased fitness can spread into the environment. NGT crops that have crossing partner in the environment, like oilseed rape, can outcross in wild relatives, and this could potentially lead to uncontrolled spread of the NGT traits or phenotypes, and may have an effect on plant diversity. And as we will also hear in the next talk, NGT microorganisms are currently developed, which colonize the gut of pollinators like honeybees. And these NGT microorganisms may be released into the environment in future, and long-term effects on pollinators and ecosystems are hardly predictable. To sum this up, in present agroecosystems and during periods of low flower availability, pollinators may rely on crops itself for pollen and nectar. Domestication and selection breeding also altered the floral chemistry with consequences for the insects. However, NGT application seemed to exacerbate and accelerate the development. Effects are particularly concerning in ecosystems where the pollinator already faced stressors such as habitat loss, and especially the release of various NGT plants in large quantities into the environment could have consequences for pollinator, and the necessary time for pollinators to adapt cannot be expected. The release of NGT crops may therefore further increase the risks of pollinator decline. The future regulation of NGT plants should therefore enable monitoring of the long-term and cumulated effects, periodical renewal of environmental risk assessment, including these long-term and cumulated effects, and an effective control of the scale and duration of release. Thank you for your attention. This is the website of the project, and we will publish the results of the study soon.
All right, thank you, Franziska. So if I understand, this will be published in a peer-reviewed journal in that case? Or, yeah.
So as a fact sheet before.
As a fact sheet, yes, okay. So for the moment, I would propose that we move on to the second part, which will deal with microorganisms derived from new genomic techniques. So on this issue, you may be aware that the Commission published in December last year a proposal where they proposed to lower the safety standards for microorganisms, NGT microorganisms, to be released into the environment. And this proposal is currently being discussed in the Council and in the European Parliament. So it seems that at Council level, the negotiations are already quite far advanced, and there will be a vote in Council on Tuesday, and there might already then be a general approach, whilst things are going a bit slower at the level of the Parliament, where the co-rapporteurs are still working on their draft report. And but the consequences of this proposal could be very, very far-reaching, because, of course, this concerns the basis of our very life and very basic functions in ecosystems. So we believe it would be very important to have a broader debate about this, to also involve the public and make them aware that this is going on. So we hope that we can be part of that effort with our webinar. And I would like now to give the word to Matthias Juhas, who has carried out a case study on a soil organism. And please, Matthias, explain us what this is about.
Yes, thank you, Astrid. We'll share the slides. And I hope you can see them.
Yes, perfect.
Okay, very good. So, okay, as Astrid already mentioned, in the last few weeks, we took a closer look at the risk assessment of genetically engineered microorganisms, because the European Commission has put forward a proposal regarding the placing on the market of genetically engineered microorganisms in December last year. I will not go into detail of the text, just very short. The purpose of this proposal is to change the current GMO legislation with the aim to create a special low-risk category for genetically engineered microorganisms in order to facilitate accelerated market access, including partially exemption from environmental risk assessment. And this reduction of safety standards is based on the classification of the parental species to be included in a list of microorganisms considered as being safe, the so-called qualified presumption of safety, short QPS list, established by EFSA. Furthermore, all authorizations are to be valid for an unlimited period of time. Methods for identification enabling monitoring are no longer to be required in every case, and the EU Commission is to be given carte blanche to change the law at any time via delegated acts. To date, there has been virtually zero experience in the EU with the environmental release of genetically engineered microorganisms. However, if the proposal is adopted, this could very soon change. And since on the 2nd of July, there will already be the deadline for tabling amendments on the proposal, I will now present some findings of our case study, which we published today. My presentation is divided into 3 parts, and in the first part, I will begin with a really short overview about a few general facts on soil organisms, soil microorganisms, which just to give you an impression why they can be regarded as natural goods that need to be protected. Starting with the interaction of soil, plants, and microbes. In this overview, it is shown that in the zone around the plant roots, illustrated here in the middle, which is also called rhizosphere, there are many microorganisms living with either neutral, pathogenic, or beneficial impact on plants. And the abundance and diversity of these microorganisms in the rhizosphere is dependent on the surrounding soil, the interacting plants, as well as biotic and abiotic factors. While the majority of microorganisms in the soil still awaits discovery, many species and strains are already described that have key functions for ecosystems, including plant and animal health. However, unlike it is, for example, the case for pollinators, there are no specific protection measures in place for this huge diversity of these microorganisms. In our study, we took a closer look at two species of soil microorganisms. The first is Bacillus amyloliquefaciens and the second Bacillus velecensis, which share many similarities, yet they are also characterized by an extremely high degree of diversity. They belong to the group of Bacillus subtilis, and together with some other species, they form the operational or subgroup of Bacillus amyloliquefaciens, short BA. In databases, there are genomes of around 1,800 different strains deposited, most of which are Bacillus velecensis strains, illustrating the high diversity even in this really small subgroup of microbes. Concerning their functions, BA microbes can also shape the composition of microbiomes in the soil as well as in animal guts, and in this context also in humans, where they can act as probiotics. As they are capable of binding heavy metals, they are also used, for example, in bioremediation of contaminated soil. In their function as biofertilizer, they can improve nutrient uptake and enhance plant growth. At the same time, they are also used in biocontrol of soil-borne pathogens. And last but not least, they can activate systemic responses to biotic and abiotic stress in plants. As I already mentioned, BA microbes can be found in a multiversum of habitats, such as the intestines of animals or in aquatic environments, but they mostly occur in the soil, where they represent a key species for plant health. And there, via secretion of hormones and volatile organic compounds, they can enhance plant growth, and through primary and secondary metabolites, they can enhance plant resistance to stress and the production of diverse antimicrobial compounds offers protection against pathogens, and the inoculation with specific BA strains significantly increase the abundance of microbes that are known to have positive effect on plant growth, nutrition, and health. After this short overview, I will come to the second part, focusing on the safety aspects related to BA microbes, and I'm still talking about unengineered native strains, just to give you a picture of what needs to be taken into account when it comes to the release of genetically engineered microorganisms, whose safety aspects must then be considered in addition, which I will show you later. Currently, BA microbes are used in two sectors. First, in contained industrial systems for the production of enzymes and antimicrobial compounds. And genetically engineered BA microbes are already being used in these closed systems. The other one is the agriculture sector, where BA microbes are used as probiotics in livestock farming and as active substance in plant protection products, mainly to treat fungal infections. Since, for example, the plant protection products are already used outdoor in the fields, there are several safety aspects that have to be taken into account in environmental risk assessment when it comes to outdoor applications. And these safety aspects result from the specific characteristics of BA microbes, with the most important being spore formation, the production of toxins or antimicrobial compounds, and the overall potential of to become an invasive species. These characteristics are relevant because the endospores of BA microbes, which, like the living bacteria, are distributed via animals, soil, water, and air, can persist and are viable for extreme long periods of time, even under suboptimal environmental conditions. Furthermore, the numerous toxic compounds have an effect on a wide variety of organisms And one interesting observation in this context is, for example, that the combined use of products containing Bacillus thuringiensis strains, which is a widely used pesticide whose toxins are also present in transgenic plants, and Bacillus amyloliquefacins result in increased toxicity in honeybees in terms of cutting their lifespan in half compared to the application of the single products. Another important aspect to consider are the signs of BA microbes to be potentially invasive species, since they are very efficient at competing for nutrients or resources. They can displace other bacteria via antimicrobial compounds, and through synergism and antagonism, they can influence or shape the composition of resident microbial communities. Following these rather general safety aspects, let's now turn to the actual handling of the so-called QPS system and environmental risk assessment on BA microbes in a concrete case study. For the BA strain IT45 applied as plant protection product, which is not genetically engineered, EFSA, in their conclusion on pesticide peer review published in 2021, could not finalize environmental risk assessment for several issues. They could not finalize consumer dietary risk assessment. They could not conclude on the persistence and multiplication of the strain in the soil, the potential infectivity and pathogenicity to non-target organisms like birds, soil, and aquatic organisms could not be conclusively clarified, and the issue of the presence and persistence of the toxins and metabolites in the environment, including the actual toxicity, could not be finally answered. The European Commission nevertheless approved the strain as so-called low-risk active substance for use in pesticide sprays, and in their final report, the unresolved issues identified by EFSA were all considered being irrelevant and dismissed. One reasoning for this decision was because the BA strain simply belongs to species that are included in EFSA's QPS list. As they write, EFSA has included Bacillus amyloliquefacin in the list of QPS recommended biological agents in 2018, where the absence of toxigenic activity is provided. So this example shows that authorizations are granted even if data gaps in risk assessment exist. And with the proposal of the Commission, the system is now also to be applied to genetically engineered microorganisms, for which significantly higher standards should actually apply. And with this in mind, I will come now to the third and last part of my talk, where I will talk about the application of new genomic techniques, which until recently were quite difficult to implement in BA microbes. And this is mainly due to the fact that wild-type strains show low transformation efficiency that has long been an obstacle for genetic manipulation of many production strains. This is caused by a restriction modification system posing a significant constraint in BA microbes, which makes their plasmid synthesis much less efficient compared to other production strains like, for example, E. coli. And this major problem was solved in 2020 by the successful application of NGTs, making BA strains accessible for manipulation, for example, as high-efficient industrial production hosts. In the following, with the help of CRISPR-Cas, for example, gene expression was altered in parallel at several sites and at multidimensional levels to increase, for example, enzyme or toxin production. Further applications are genome-reduced strains lacking, as they call it, unnecessary DNA that show faster growth rates, better transformation efficiency, and a higher expression capacity. And these strains could then serve as a chassis for various industrial purposes. What most of the current NGT applications in BA microbes have in common is that they enable complex metabolic engineering without introducing any additional genes. And this is really important for the application of the QPS system, since in this concept, to get on the QPS list, the microorganism is not allowed to have so-called genes of concern, like resistance genes against antibiotics or genes involved in toxin production. To derive to these NGT BA strains, it is sufficient to knock out genes or causing overexpression of other genes, both simultaneously and at multiple sites. And the resulting patterns of genetic changes of these NGT BA microbes are unique for NGTs, and the characteristics introduced by them are new to nature. Consequently, genetically engineered BA microbes can have a much higher potential to become invasive or toxic to non-target organisms, which can have serious consequences for crop and wild plants, pollinators, microbial communities, or humans. Coming to the conclusions, given the complex functions and the key role of native BA populations, the release of genetically engineered strains into the environment should go along with highest precaution and, in case of doubts, should not be allowed for several reasons. First, if the functions of natural BA populations are destabilized or key interactions are disrupted, this may cause chain reactions in the ecosystems with potentially detrimental consequences if, for example, they are irreplaceable by other taxa or replaced by less efficient ones. And this implies, secondly, that the extremely valuable ecosystem services provided by BA must not be put at risks by introducing NGT strains that are new to nature. Furthermore, since NGT BA show characteristics that cannot be obtained from previous methods, even if no additional genes are inserted, but rather manipulated with knockout or alteration of gene regulation, they have to be considered as new to the environment. And if for these NGT microorganisms, the QPS system will be applied, this will allow to authorize the release of genetically engineered BA microbes into the environment, even if, due to major data gaps, risk assessment cannot be finalized. And this would mean a very drastic lowering of safety standards compared to current GNO regulation. And this shift towards lower safety standards could potentially be even more problematic when considering that the QPS list also includes many fungi, algae, and also viruses. And finally, it is essential for genetically engineered microorganisms belonging to species that play a role in soil and gut microbiomes to undergo a case-by-case environmental risk assessment, as well as case-specific monitoring, as currently requested under 2001/18. And in respect to the current state of knowledge, the QPS concept, which was established for food and feed and not for environmental release, cannot be applied or is not suitable to replace these assessments. We therefore propose that potential releases have to be treated with high caution, and in case of doubt, they cannot be allowed. And with this, I'd like to thank you for your attention and look forward to the discussion.